Hello everyone, welcome back to my channel. I hope you all are doing well and you are preparing pretty well for the DBT-JRF examination. So I have previously uploaded so many videos. You need to watch them out in order to gain insights of what can be asked in your DBT-JRF examination. So do watch those videos. I will provide the link in this video description box. You can check it out. If you like my effort, you can subscribe the channel for more such content, and it also gives me motivation to make more such content. So this is the first question. The relationship that involves the exchange of nutrients between two different species for their mutual growth is called what? यानी कि organism A है and organism B है. There is a mutual exchange of nutrients between two different species. Suppose ये species एक किसी bacteria का है, bacteria 1, and this is the bacteria 2. Both are two different species. So what is this known as? Antagonism? No, this is not antagonism. Commensalism? No. Parasitism? No, it is syntrophism. So syntrophism is the correct answer for this question. Syntrophism, also known as syntrophy or cross feeding, is a biological phenomenon where two or more metabolically different organisms cooperate to perform a metabolic function that none of them alone can do. यानी कि organism A and organism B दोनों mutually interact करते हैं for sake of their metabolic नीड, यानी मेटाबॉलिक नीड जो वो अकेले पूरा नहीं कर सकते हैं, जब वो साथ में मिलके अपना मेटाबॉलिक मशीनरी को चलाने के लिए जब एक म्यूचुअली वो इंटरैक्ट करते हैं, दैट इज नोन एज सिंट्रोफिज्म. नाउ नेक्स्ट क्वेश्चन. Which of the following bacteriophages has a genome composed of single stranded circular DNA? So आपको ये बताना है which of the following bacteriophages has a genome composed of single stranded circular DNA. So it is the phage 174 which is the correct answer for this question. So इस टाइप का हमेशा पूछा जाएगा कि किस बैक्टीरियोफेज में, नॉट जस्ट बैक्टीरियोफेज बट ऑल्सो अ वायरस का भी पूछ सकता है कि जेनेटिक मटेरियल सिंगल स्ट्रैंडेड आरएनए होता है या सिंगल स्ट्रैंडेड डीएनए होता है. So you have to remember all these things. So here is the classification: some viruses that infects vertebrates. So out of this table you can see DNA viruses with envelope and DNA viruses with no envelopes. So firstly you need to know that whether the genetic material is DNA or RNA, and after that whether it is enveloped or whether it is non-enveloped. So in the DNA viruses with envelope, pox viruses are there. Pox viruses include smallpox virus, cowpox virus, monkeypox, and it has double stranded DNA. And if we talk about the herpes viruses, which called cold sores by herpes simplex virus type 1, genital herpes, STD, and apart from that chicken pox and shingles. So infection, mononucleus and Burkitt lymphoma all are caused by this herpes viruses, which has again double stranded DNA, and it is an enveloped virus. So you have to remember this example: pox virus as well as herpes virus. Now DNA viruses who do not possess any envelope, like adenoviruses, papovaviruses, and parvoviruses. So these adenoviruses have double-stranded DNA. This papovavirus also has double-stranded DNA, but this parvovirus has single-stranded DNA. So always the examiner will ask you the odd one out thing, like the parvovirus. So parvovirus is a single-stranded DNA virus which requires a helper virus in order to multiply. So papovavirus, what does they cause? They cause human warts and some degenerative brain diseases, some kind of cancer, including the cervical cancer. So adenoviruses. So adenoviruses are known for the respiratory tract disorders like sore throat, tonsillitis, conjunctivitis, and gastrointestinal disorder. And there are more than 40 types of adenoviruses that are known to cause disease in humans and also in animals. So they have also the double-stranded DNA. So you have to remember the DNA viruses with envelope and without envelope, and which of the following has single-stranded DNA. So parvovirus is the group of non-enveloped virus in which it has single-stranded DNA as its genomic material. Now after this, RNA viruses with envelope and RNA viruses without envelope. So in this RNA viruses with envelope, so toga viruses cause rubella disease which has single-stranded RNA. And in orthomyxoviruses, they have also the single-stranded RNA and they cause the influenza. Like paramyxoviruses, they are responsible for the measles and mumps in humans, and they have also the single-stranded RNA. Rhabdovirus, which is responsible for the rabies, they have also the single-stranded RNA. So all these RNA viruses mentioned in the first half of the table has single-stranded RNA. You can see here, like coronavirus. So this is the recent pandemic pathogen that is responsible for causing the severe acute respiratory syndrome, upper respiratory tract infection. So it has also the single-stranded RNA. Flaviviruses, they are known for the yellow fever and West Nile virus, hepatitis C kind of diseases. And filovirus is responsible for hemorrhagic fever. And Bunya virus also has single-stranded RNA. Retrovirus, the causal organism of acquired immunodeficiency syndrome, यानी HIV कराने वाला retrovirus, उसके पास भी single-stranded RNA होता है. So these are the RNA viruses with envelope category, and in RNA viruses without envelope category we have picorna viruses. In picorna viruses we have polio, hepatitis A, and in picorna viruses single-stranded RNA genome is there, and in Rio viruses they have double-stranded RNA. So in the examination, odd one out will be asked by the examiner, like which of the following RNA viruses has double-stranded RNA in it. So it is the Rio viruses. Like हमने DNA में देखा था कि parvovirus जिसमें single-stranded DNA होता है, likewise RNA viruses without envelope में Rio virus is the virus which has double-stranded RNA in it. Now moving on to next question. So this is the next question. In which of the following is the basic principle of Sanger's DNA sequencing method? So Sanger's DNA sequencing method is also known as the chain termination method and also the dideoxy method, because in this DDNTPs are used, that is the dideoxy NTPs. So this method is known as the chain termination method by the incorporation of dideoxynucleotide. जब भी growing chain में DNTPs की जगह DDNTPs आते हैं यानी dideoxy, so chain termination होता है because it lacks the OH group, and उस वजह से क्या होता है, chain जो है वो further elongate नहीं हो पाता है because of lacking of that OH group. So the correct answer for this question is option A. Now moving on to the next question. An element that is present in the nucleotide but not in a nucleoside is of course the phosphorus. So this is the basic question. DBT में ये सारे questions भी आपको मिलेंगे, A section में देखने को मिल सकते हैं. The basic difference between nucleotide and nucleoside is the phosphorus. So the right answer for this question is phosphorus. अगर हम लोग बात करें Krebs cycle की, so in this Krebs cycle like TCA is dash pathway. Tricarboxylic acid cycle is a dash pathway, an anabolic, and catabolic, amphibolic or pyogenic. So you can see if a pathway has both catabolic and anabolic phase, it is known as amphibolic. So the right answer for this question is an amphibolic. TCA is an amphibolic pathway because it has both anabolism as well as catabolism. In anabolism, the TCA cycle generates precursor for biosynthetic processes such as formation of fatty acids and amino acids, and in the catabolic pathway it oxidizes the acetyl group to produce carbon dioxide and NADH and FADH2. So you can see it has both the synthesis of material, that is the anabolic pathway, and the oxidation or the degradation breakdown of a substance, that is the catabolic pathway. So if a process has both anabolic and catabolic, that is known as the amphibolic one. So TCA is a amphibolic pathway. Now moving on to the next. All pseudogenes do not code for a protein with original function, protein with altered function, RNA with coding sequence, RNA with regulatory function. So it is the protein with original function. So the right answer for this question is protein with original function, because all pseudogenes are similar with the original genes, but their protein coding capability is somehow altered because of some kind of mutation in it, so it is not able to synthesize the actual protein which is required. So all pseudogenes are more or less like a normal gene, but they have a faulty protein coding section in it, so it will not generate protein with its original function. So the right answer for this question is protein with original function. Now a matching, which is the very important, in which you have to mention the biological vector of each disease. So there are so many diseases mentioned here. So you always have to find the option which you are familiar with. So the first one is leishmaniasis. So this leishmaniasis is also known as the Kala Azar. So this is the Kala Azar. So Kala Azar is transmitted by a sandfly, bite of a sandfly, yani ARCA3. So ARCA3 is in this option and in this option. So you should eliminate the second one and the fourth one. Now, Chagas disease. Chagas disease is also called the American trypanosomiasis. Trypanosomiasis, which is caused by the Trypanosoma cruzi. So this disease is just transmitted by the redwood bug, which is also known as the kissing bug. So if in question it is asked kissing bug, then also it is right, kissing bugs. इसी को ही बोलते हैं kissing bug. So P का 4 के साथ होगा matching. So P का 4 is only in first option. Then you can eliminate this third one also. Now trypanosomiasis. So this is also known as the sleeping sickness. Sleeping sickness. So this is caused by the bite of tsetse fly. यानी Q का first. तो Q का first कहाँ है? यहाँ है. Now yellow fever. Yellow fever is caused by the bite of the mosquitoes. So S का 2. So yellow fever is caused by mosquito. So the right answer for this question will be option A. You can see. So you have to remember the common name of the disease also, the causal organism also, and the vector also. So from this 3 type of questions can be asked in DBT or any other exam. Like you can ask the common disease of the like you may ask the common name of the disease, the causative agent, that is the pathogen name, or from which vector it is transmitted. So you can see how many questions can be arise from this single question. Now moving further, this one. So you have to again match the industrial enzyme with its application. So you must be familiar about the lipase, fican or ficin, amylase and glucosidase. So out of this, you must be familiar with this lipase, which is a lipid hydrolytic enzyme. It basically degrades the lipids. So lipid means oil. So it will degrade the oil. So for P का matching, first का matching second के साथ हो रहा है. तो P का 2 किसमें है? P का 2 इसमें है and इसमें है. So you can eliminate A and you can eliminate D. Now apart from that, amylase. So amylase is responsible for the maltose syrup production. Maltose syrup production में responsible है. It is an enzyme which is responsible for the cleavage of alpha 1,4 glucosidic bond that generates short chain oligosaccharide like disaccharide and also some other oligosaccharides. So maltose syrup is readily produced by the amylase enzyme in industries. So R का matching होगा first के साथ. तो R का matching कहाँ है? दोनों में ही दिया है. तो आपको उन दोनों को भी consider करना पड़ेगा. Now glucosidase. So glucosidase is an enzyme responsible for the generation of oligosaccharide as well as monosaccharide by hydrolyzing the glycosidic bond. So glucosidase will be matched with this option 3, S ka 3. So S ka 3 is only in B option. So C भी eliminate हो गया. And this ficin is a protease which is responsible for hydrolytic action on proteins. So it will basically degrade the protein, hydrolyze the protein. So it is used for the meat tenderization by the proteolysis action. So ficin का matching होगा 4th के साथ. So Q का 4 किस में दिया था? इसी में दिया था. यानी correct answer हमारे इस question का है option B. So all this can be asked individually also. So you have to remember all these 4 options, the industrially important enzymes. Now next question. So this is again matching question. You have to match the column 1 with its corresponding use. In name itself, you can suggest that glutamine is an excellent source of carbon as well as nitrogen in the animal cell culture media. Glutamine. So P का matching होगा 3 के साथ. P का 3 किसमें किसमें है? इसमें है और इसमें है. So you can eliminate the B and the D. Trypsin is a protease which is used for the detachment of adherent cells. So ये directly match हो जाएगा इसके साथ में, Q का matching होगा first के साथ, Q का first is in A option also and in C option also. Hypoxanthine. So hypoxanthine is nothing but a component of medium for selection of hybridoma monoclonal antibody production. So hypoxanthine का होगा ये वाला. So basically hat medium, that is the hypoxanthine, aminopterin and thymidine. So this is the selection medium for the antibody production. तो hypoxanthine का matching होगा 4 के साथ, यानी R का 4 किस में है, फर्स्ट ऑप्शन में है, यानी C भी एलिमिनेट हो गया. Neomycin is an antibiotic which is used for the selection of transfected mammalian cell lines, यानी इसमें. So the right answer for this question will be option first, that is the A option. Now one more question from which questions can be asked, that primase, reverse transcriptase, RNA replicase and DNA polymerase 3. So you know primase is a very crucial enzyme for the replication process of DNA. So it generates short chain RNA primers. So it is based on the DNA dependent RNA synthesis. It synthesizes short chain RNA primers. So P का matching 4 के साथ होगा. तो P का 4 यहाँ दिख रहा है and यहाँ दिख रहा है. So eliminate both B and C. Now reverse transcriptase. So reverse transcriptase is known for the synthesis of RNA from a DNA template. So Q का matching होगा 3 के साथ में. So Q का 3 हो गया, यानी कि Q का 3 किस में दिया है, सिर्फ 4th में दिया है, तो ये भी एलिमिनेट कर दो. Now RNA replicase. So RNA replicase is responsible for the RNA dependent RNA synthesis, यानी R का first. Now S का क्या है, DNA polymerase 3, S का 2. So DNA polymerase जो है, it is responsible for the synthesis of DNA based DNA synthesis, यानी DNA dependent DNA synthesis ये कराता है. So the right answer for this question is option 4. So see कितना important question था ये, आपको A section में DBT के and also in B section पूछा जा सकता है. So this is all for today. I hope you liked it. Thank you so much for watching and do subscribe the channel for more such videos.